My name is Dr. Vassie Ware, and I want to tell you a little about the research that we do here in my laboratory. We have two basic projects that we're working on. One is to study ribosome heterogeneity in Drosophila, as well as ribosome-mediated gene expression and regulation in Drosophila. The second project is a phage biology project in which we study antiviral systems that are encoded by phages. I want to tell you a little bit more about what happens here in my laboratory with some slides. So hold on for just a minute.
Good afternoon, I'm back. I wanted to be able to share just a few slides with you so that you could get a little visual of some of the work that we do in my laboratory. As I mentioned, we study ribosome heterogeneity at the protein level, and there are some very specific questions that we're interested in. If you take a look at the left side of the slide, you will notice that there are a series of questions that are related to ribosome heterogeneity. But the questions that we are particularly interested in are encircled in red. The first being, how do specialized ribosomes recognize their targets? And immediately you are probably wondering, what are specialized ribosomes? And I'll tell you a little bit about that momentarily. But one of the other questions that we're interested in, as shown here on the left, is, are there cell-specific types of ribosomes? And so first of all, let me say to you that by virtue of the fact that we're going to talk a little bit about specialized ribosomes, that indicates that not all ribosomes are obviously alike. On the right-hand side, you will see that there is a diagram essentially of a bacterial cell that has been infected with a bacteriophage, and that phage genome has in fact been integrated into the genome of the bacterial cell. And what we find as a result of this is that the phage, which is then called a prophage, has the ability to alter the physiology of the bacterial host. As a result of that alteration in host physiology, you sometimes find that that bacterium, which now harbors a virus in its chromosome, which is now called a lysogen. That lysogen has the ability to ward off attacks by other incoming viruses. And some of that ability to defend itself is actually encoded by genes that are in the prophage. And so that is another area of interest in my laboratory. Let's talk a little bit about ribosome heterogeneity in more detail. And I want to tell you that the family of proteins that we study for heterogeneity is a family that's called the RPL22 family. The model organism that we use is Drosophila melanogaster. And I want to show you in this model of the large subunit where L22 or L22-like, which are the two members of that ribosomal protein family, where they would be located at the base of the large subunit, as you might see my cursor showing you here. Now, what's interesting about this is that either of these proteins can be incorporated into the ribosome itself, but they can't both be incorporated at the same time. And so by virtue of a mutual exclusive assembly, we in principle could have two populations of ribosomes in a cell if both of those genes were in fact expressed. And over on the right, you can see what we have come to find out by studying the expression of these two proteins in the testis in Drosophila is that L22 and L22-like are both expressed in germ cells. Only in the somatic cells of the testis do we find L22, but L22-like is a germline specific ribosomal protein within the testis. We are also studying L22-like expression in some other tissues, and I show on the left here expression of L22-like within the developing eye of Drosophila. And this is an image that shows you in green a set of cells that we call interomotidial hair cells. They look like little bristles that project from the surface of that compound eye. And you can see here, as depicted in green, that L22 is shown, L22-like rather, is shown here as these little green images that project from the surface. On the right-hand side here, I show you an image of image from the ovary in Drosophila. And in the ovary, we find that L22-like is expressed in germline stem cells up here in this part of an ovariole, which will happen to be the home for developing oocytes. And so the more mature oocyte is follicle is here with the oocyte depicted here at the very bottom. But as you can see, only in the germline stem cells do we find expression of L22-like. And if you recall, in the testis, we were able to show that this, this protein is expressed in all germ cells. So you may ask the question, if we actually have two populations at least of ribosomes within the testis that are different with regard to their protein level at the L22 or L22-like stage, what's the purpose of that? What can we distinguish any features of those ribosomes that might be important with regard to function? And so we undertook a couple of years ago, an RNAseq study whereby we were able to specifically isolate L22 containing polysomes and compare them to L22-like containing polysomes. And what we were able to then do is to isolate the RNAs from those polysomes and determine that there are specific types of mRNAs, and in fact some other classes of RNAs as well, that are dedicated or enriched on particular ribosome types. And so this is a very interesting phenomenon where we then can talk about specialized ribosomes because there are different populations of mRNAs that are in fact translated on different ribosome types. And so this is an area of interest, great interest in my laboratory. And now we are trying to uncover the features of the mRNAs or features of the ribosomes themselves that are targeting or allowing for the enrichment of these mRNAs. So that whole specificity of how a particular messenger RNA is recruited to a ribosome is of great interest to us. And then the other major interest in my laboratory is to study phage genomics and phage encoded antiviral systems. And as I mentioned at the beginning of this discussion, there are phage genes that when they are part of the genomic profile of a lysogen, those genes can sometimes give a defense characteristic to the lysogen as by virtue of that virus actually expressing particular genes. And so we've been studying a mycobacteria phage called Butters, named by one of the undergraduates who in fact discovered this. And that mycobacteria phage, when it is a prophage, gives the lysogen particular characteristics of defending against attack by other types of viruses. And so without too much more detail, we are working on a model to understand how particular genes that are in the genome of Butters, in fact, convey that particular defense characteristic. And so there are several genes here that you can see in this model that we are studying, and there are any number of features of this that are important because we want to understand how these, not only how these proteins, how these genes work, but we want to then understand what the possibilities are for thinking about applications of this type of work to features such as phage therapy, or thinking about how to eradicate bacteria that are perhaps resistant to antibiotics. And so these are some of the features of work that we do in my laboratory. And I wanted to just show you the current members of my lab. I have a research associate who was a former PhD student in the laboratory, 2 additional graduate students, a series of undergrads who do a lot of important work, and recent PhDs, Brett Gershman, Katie McGeaney, and Mike Kearse. And Brett is still in the laboratory, as I said, as a research associate, but Katie and Mike have moved on to important work that they are doing in jobs either at national labs for Katie, and Mike is now an assistant professor at The Ohio State University. And so